hyphae is delimited from the remainder of the hyphae. Okay, so basically, as I mentioned to you here, if this is the hyphae here, like this, become like that, and this one is becomes like this. Okay, will become like this. Okay, now delimited, that means it becomes like this. Okay, so now when one becomes like this, now this is known as gemitangia. Do you see that? Okay, as I already showed. Okay, to mention here, the delimited portion is gemitangia. Can you read it here? Known as gemitangia. Okay, and subtending hyphae if I put it down here, is the suspensor, known as suspensor. You feel? So basically, if I put it down here, I hope I'm not confusing you. If this is the gemitangia, this is going to be the gemitangia here, and this is going to be the suspensor. Do you follow that? Okay. All right. Yeah, because you see early September, okay. So simply the third stage is going to come after these two gemitangia make contact, they're going to dissolve. So the other point I'm going to mention after the gemitangia, okay, have made contact, the wall or the cell wall between, okay, the tips, okay, of course, between the tips of the two gemitangia, don't forget that, between the two, okay, between the tips of the gemitangia, dissolve. Okay, dissolve. And all what happened here, and the cytoplasm and the nuclei, there are many nuclei here, okay, called lie free within a single lumen like a chamber. You follow that? So basically, when these two hyphae, it becomes, say, like this one here. Okay. So they become dissolve and become like called lumen like this. So all the cytoplasm become there here. Okay. Like free within a single lumen. Okay. when that happened, then during this period, okay, put here the nuclei, okay, the nuclei, and it's called the uniting gamitangia, and the uniting gamitangia, okay, increase, okay, increase in number, okay, and as mentioned here, of course, and the plus and minus the nuclei will unite, okay, right, of course, forming many zygotes. In this case, of course, the zygote is becoming diploid, okay, and the, okay, and we call that chamber there, and the chamber, okay, which will be enclosed, okay, by a thick wall known as, okay, zygospore. Okay, and as I showed you here, I guess, yeah. Okay. As you can see here, that basically united thick wall, and it's known as the zygospore. Okay, then finally, the germination of zygospore, okay, when the zygosporangium, when the zygosporangium split open, okay, split open, all right, and as meiotic division occurs, sorry, in the zygospore, okay, then the resulted those are haploid and they will germinate into a new, okay, hive or to new organism. For that, and then of course, and the life and the life cycle repeat itself. Okay, if the conditions allow. If the conditions allow. Okay.
Can you move the paper left higher?
Okay now. Okay. Now, what I'm going to mention to you here. Okay. Now, usually, just give me a second here. I will put it down. When these two hyphae they come together, it seems the studies has shown there is a kind of chemical stimuli we call it, which is produced by the hyphae to bring them together, known as a progamon. So we're going to mention this point at the end here. Okay. Now. Okay. It has been shown. Okay. Okay. That that the progametangia okay do not develop okay if the mating types if the mating types are grown separately. That means if they grow the two culture from each other, unless if these together, and that suggests there is a chemical signal there, okay? And thus, it has been, we call it postulated that, okay, called progamon inform the compatible inform the compatible, because that means plus and minus, what compatible hyphae or mycelia.
Sir, could you move your page up a little bit? Thank you.
Okay. And from the compatible mycelia. Okay. Of one another proximity. Of one another proximity. Okay. For that? However, I'm going to mention here one point, okay, that the same what happened here when the hyphae they come together. This is what you call the progamon. Okay, basically what it is basically the hyphae will secrete a kind of acid called what they call a trisporic acid. Okay, this is kind of trisporic acid. Now, the hyphae of a closed, they secrete that trisporic acid, and that's basically allow when the hyphae or the mycelium in a proximity will raise and form that progamitangia and until they reach together and touches and the wall dissolve. Is that clear? Okay. So it shows that progamon basically is a kind of chemical, okay, which is being secreted by the hyphae known as trisporic acid. Okay. So basically, this is the sexual life cycle of Rhizopus, and already we mentioned about the okay. And now I'm going to basically to talk about another example. Okay. Still, just for this one, just in case somebody didn't finish it. Okay. Okay. I'm still basically with the class zygomycetes. Okay. I'm still also with the order Mucorales. Okay. And the example I'm going to use at the moment, just a kind known as a fungus known as Bilobolus christianus. This one here, called Bilobolus christianus. You see it? Okay. Now, these fungi, basically what they are what they call, we're known as coprophilous fungus, which is basically what we call dung inhibiting fungus. Okay. And basically what it is, this fungus, I'm going to give you an example, when they grow in the dung, especially like rabbit dung or so on, okay, then they will germinate. Just give me a second here, we'll take this out now. Okay. They will germinate, and usually each of these fungus has what you call the sporangiophore is very long. This sporangiophore is almost like sometimes it reach about 2 to 4 centimeter in length, and basically they develop here what you call like a small vesicle. This is the sporangium here, and under the sporangium we call subsporangial vesicle. And the subsporangial vesicle, what happened here, it later on it built a pressure, and basically that fungus, the sporangia, it has like a retina, which is basically stimulated by the lights. And unfortunately, sometimes if you do it in the petri dish, as you used to do it in the lab, and when you have to, you find all these sporangia hitting to the light. So once you come to the light, that pressure, as a matter of fact, it reached sometimes to 100 pounds, and then it would shoot with a spore to about 1 feet or a couple of feet away. Now, the idea is, of course, when this fungus grows in the dungs of rabbit or other organism, okay, then when they shoot the spore, basically they will attach this spore to another nearby plant or a leaf or anything else. And so that basically in the end, when the animal will eat them, okay, they will cycle, what you call, will repeat itself. Is that clear? Okay, so that's the example we are going to do with this about this called Polybolus carcinalis. Now, so to write few lines only on this, I'm not going to give the life cycle, but just to explain what already I mentioned to you. So that the first point I'm going to mention here, basically this is a typical, this fungus, it is a typical, okay, called coprophilus. Called coprophilus fungi, in other words, known as dung inhabiting. Okay, it is dung inhabiting. Okay, which is also belong, the species belong to the order Mycorrhizas. Okay. This fungus, it has, okay, it has a sporangiophore, it has unbranched, as I showed you, unbranched, known as unbranched sporangiophore. Okay. About 2 to 4 centimeter tall, and as I mentioned, and have a unique explosive dispersal mechanism, a unique explosive, okay, sorry, a unique explosive dispersal mechanism. Okay. Now, the other point I mentioned, the whole sporangiophore is a single cell. Like basically all that is a single cell. All that you can see, all this one is single cell. And if you enlarge it, basically it's going to look like this one with the pressure here. Okay, that is basically how it's going to build a pressure here. Okay, we're going to talk about that. And to mention a few lines here. Okay, now the whole sporangiophore means the whole cell is a single large cell. Is a single large cell. Okay. Now, as you can see here, beneath the black, okay, beneath the black, okay, you can make a sketch yourself over here. The black apical mytosporangium, I'm going to continue here. Mytosporangium, that means sporangium in which the spore are developed by mitosis. Mytosporangium, mytosporangium. Okay, that black apical mytosporangium is beneath it is a lens like, okay, subsporangial vesicle, sporangial vesicle. Okay. And this is basically with a light sensitive retina. 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 And this retina basically that controls the growth, okay, of the sporangiform of of the sporangiform, okay, very precisely because it's directed toward the light, very precisely, sorry, okay, aiming it accurately toward, okay, toward any light source. Okay. In other words, it is phototrophic. That means really organize itself. In other words, it is phototrophic. This is R here, phototrophic. Okay. And just to mention here, basically that the subsporangial vesicle, this one here, when we mention the subsporangial, okay, the subsporangial vesicle, okay, able to build, okay, pressure here up to almost close to about 100 pounds, up to 100 pounds, okay, per square inch. Pound per square inch. And that's basically when it's explode able inch, okay. And this eventually, okay, causes the vesicle to explode. And by doing so, of course, to explode, okay, ejecting the sporangia, ejecting the sporangia, okay, sometimes to a distance up to sometimes 2 meters. Okay. So basically, the idea it is basically because that spore, the wet fungus grows in the dung. So the idea it is basically to raise and then to shoot this spore to nearby any plants here and there, so that basically the animal will eat it again. Of course, this fungus does not cause any harm, and even the fungus will stay all the spore alive, even when it passes through all the digestive system of the animal. Okay, just to mention a few lines here. Okay. And basically, as you can see, the sporangia here, from its shape, you can see basically it has like a sticky end. So it makes it very easy after being exploded to be what you call to stick to nearby plant or so on. Just to mention this point here. Okay, just yeah, make sure that okay. Then the sticky sporangia, or sporangium in this case here, the sticky sporangium. Okay, as a matter of fact, okay, adhere. I'm going to mention here to nearby leaf or plant. Plant that will be eaten by the animal. Okay, to repeat the cycle. To repeat the cycle. See that? Okay. So basically, this is another example, as I mentioned to you here, of the of this what you call the dung inhabiting kind of fungi. Okay, which is known as Polybolus christianus, called Polybolus christianus, as I showed to you here. Okay, called Polybolus. Yeah. All right. All right. I think I gave you in the zygomycete, what you call in the zygomycota, the phylum, for what you call in the zygomycete, we have a class zygomycete. We have 4 orders. Do you remember that? We have one order of Mycorrhizal. Now there is the other order. I'm going to, okay, known as the order. Put here class zygomycetes. We have the first order, which is give you the order Mycorrhizal, which is the most common one. Now this order here, the second order here, is known as order Entomophthalus. Entomophthalus. Okay. And basically this one include the species known as fly fungi. Sometimes basically if you work, for example, in closed windows, and especially if the windows has not been cleaned, you find there is a lot of flies around. It's dead. Basically, if you take any of these flies and you examine it under the microscope, you can see in the wing there is like a very wide kind of fungus. So this fungus, as a matter of fact, infect the wings and kill the flies. Okay, and that's called the fly fungi. And that's we call the fly fungi. Okay, and the example maybe also they give it to the lab to the slide known as Entomopthora musica. The example known as Entomopthora musica. Okay. Just a few lines I'm going to give you in this one. As I mentioned to you, basically these fungi, simply to mention, are known or are so called, whatever you want to put down. Okay. The fly fungi. Okay. Which attacks and that's why basically some of these fungi could be used. There is a research try to use basically like insecticide to become like what you call like a biological control for insect and other pests. Okay, so these fungi are so-called fly fungi because simply which attack, as I mentioned you, flies, as you can see, flies can be seen on long unwashed windows. Okay, like for example in garages, any places. Okay, unwashed windows to mention to you. Now these fungi, basically, it is really, um, uh, of course, you didn't see it. I don't know if they show you a slide of the fungus. Basically, it looks like a white if you have the insect. Okay, supposedly this is an insect here. I'm just going to give you whatever an insect you have here. And this is the wing. Usually it grows here and form like a white thing. So basically the fungus itself, it has like a very small conidiophore and form like a small conidia, small spore. This is how it looks like. Just like this one here. Okay, you see how it's put only one conidia at the top here. And that's basically when they grow between the wings, they will form many conidioform with the wing. And that basically it looks white because the color of these small spore, which you call conidia, is also white. Just to mention a few lines here. Okay. Now, as I mentioned to you here, that these fungi, okay, shoot their one sporangia, one dash here, one spored sporangia, okay, of the tips of the sporangiophore of the sporangiophore. Okay. And that's why basically to mention this point here, to mention that an infected fly or insect and infected fly or insect, okay, by this fungus, okay, shows a wide zone which consists of thousands of conidia. Okay, because these conidia, as I told you, it expels out of the sporangiophore. Okay. Now, the last thing I'm going to mention this fungus, because of this ability of this fungus to kill fly, so there is an idea just to use it, what you call in biological control. just to mention. And this species of Entomophthora, to mention, and this species of Entomophthora, okay, are being investigated. Okay, are being investigated for their potential use as I mentioned to you, in the biological control of insect pests. Of insect pests. Okay. Before that, okay. Then I'm going to move to the still the third order of the zygomycetes. The same thing we have here, the class zygomycetes. Now, this order known as Kixeliales. Order. Okay. Okay, this is K. Zelialis. I'm just going to put only a few lines. I'm going to talk to you about here the example. Okay, the example known as Comencia of the genus Comencia. Okay. Because this fungus basically grows on kind of insect, but has been basically taken from, found on bat dung from a cave. Okay. Now, I'm just going to read this one. I think easily you can follow it. Just to mention that members of this group are typical zygomycetes. Okay. Members of this group, because I'm going to write to you because there is Sporangius has a lot of different names, and that's what I'm going to, these are the one I'm going to write. Okay. But the few line, the first one, a few line, very easy to follow. That members of this group are typical zygomycetes. Okay. And they have regularly septate hyphae, this fungus, and they have regularly septate hyphae. Now, I'm going to give you here, you have to come across it in one way or another. Two terminology. Just now we have telomorphs and anamorphs. Anybody came across this term? Telomorphs and anamorphs? Definitely not. Okay. When you say telomorphs, just a minute here. sorry, teleomorphs. Oh, sorry. Okay. Teleomorphs. Okay. Basically, that's when you talk about their sexual production. Because sometimes if you read some text or some book, you may find these terms. So it's better to know. So we say telomorphs, that means their sexual reproduction, sexual cycle, whatever it is. Or when we say anamorphs, okay, this is the asexual. Okay, we'll try to point to you that these fungus basically they don't have much knowledge yet about their telomorph, that they don't have much what you call idea about their sexual manifestation, though they have a little bit more about their asexual manifestation. Okay, and basically they produce quite kind of complex type of spores and conidia. Okay, so as I mentioned to you, very simple, going to repeat the sentence that these fungi, their telomorphs or sexual manifestation are unknown, but their anamorphs or asexual manifestation, okay, known and occur by a production of conidia. Okay. Before this, now, what I want you to read, to focus, do you see this figure?
Yep.
Do you see the writing clearly, or I want to enlarge it for you? Okay, now, because I want to mention, because this is basically is the sporangiophore, that is the structure of the sporangiophore. You have the axis, and then you have this. So I'm going to describe there, this one. Now, and this comments here. Okay, so I will show it to you each time. Now, it's a sporangiophore. It's of this species here. It's sporangiophore. Okay, tall and bear many fertile side branches, many fertile side branches, many fertile side branches called, okay, I'm sure that you see it, called sporocladia. Sorry, do you see this called sporocladia? ADIA, sporocladia. Before that, called side branches known as fertile side branches known as sporocladia. And each of these okay, produce a row of lateral cells. Produce, okay, a row of lateral cells called pseudocladia or pseudo. I think put it down for you there. Pseudophladius. I'd like to mention that word there. Known as pseudophladius.
Sir, could you adjust your page once again, please? Thank you.
Okay. Called pseudophladius. And from the apex of this one here, and from the apex, okay, from the apex of each of pseudophaladia, or pseudophiladius, arises and elongate when spored, mitosporangia, when spored mitosporangia. Okay, just give me a second here. I'm going to show this here. Now, did you see this figure here now clearly? Okay, this is the axis of the sporangia. Now, it become these are the side branches, which is known as sporocladia. Do you see it? Yes or no?
Yep.
Okay. And then out of these sporocladia, it become the side branches like this one here. Okay. Okay. Now, these side branches, you see, if you look to this one here, I'm going to put it down here like this one here. Okay. Because these are the sporocladia, and then you find a small like this coming like this one here. You see this one here? Yeah. Okay. And this one known as pseudo. I'm going to mention just, I will show it to you in a second here. Okay. Phaladia. Okay. And then here becomes spores. Okay. So basically like this one here, as you can see, that these are the sporocladia. Okay. Now these, the tall one, it comes like this one here. This is known as the pseudophaladia. And then at the end, you have a long sporangium here. So that basically what shows to you here. Okay. So basically this is how it looks like. Okay. This is the sporocladia. Okay. And this tall one here, known as the pseudophiladius. And at the end here, you find there is a small kind of spore, which is known as what you call elongated one. Okay. That's why basically it shows that definitely it's quite complex way of forming these kind of asexual kind of spores. Okay. So, okay. So basically I just go back here what I showed to you, that each of these produce a row of lateral cell called pseudophilades, and of course sporangium basically that there are many side branches known as sporocladia, and then basically each of these sporocladia bore what you call pseudophilades, and then these at the end develop what you call one spored mitosporangium. Okay, so all this is what I want to talk to you about, these kind of Cucuzalis, which is basically found, as a matter of fact, in some of these bat in some of the dung, bat of the dung. Yeah, they took the dung basically from the bat and they find it growing there from the kid. Then going to move to another order now. Time goes very quickly here. Oh, because you have quite a bit of material anyway. Okay. Now still with the class Zygomycete, but here we have the order known as Glumielis, and this is very important order called order Glumielis. Okay? Because that order basically includes some of the fungi which of the species which form what you call the mycorrhizae, particularly the endomycorrhizae. Okay, now, so to mention a few lines of this one here. So basically, this fungus, as a matter of fact, is soil inhabiting fungi. Okay, now, these, okay, soil inhabiting fungi, okay, though basically for some reason they put them in zygomycete, but it seemed till now they didn't find any of these produce zygospore. And that's why I'm going to mention this sentence for you, that these soil inhabiting fungi were placed, okay, in the zygomycetes, okay, and the zygomycete we call tentatively. It was important to know that because since none of them form zygosporangia, since none of them form zygosporangia. This is number one. Now, as I mentioned to you here, okay, that these are very important because these fungi are very important because their spores or their for what you call the mycorrhizae. To mention this point here, okay, these are extremely important. These are extremely important important because their hyphae, okay, enter the living roots, because as I mentioned to you before, mycorrhizae is very important for the growth. Almost 90% of all higher plants, they should have some sort of mycorrhizae in their roots. And the importance of that, what it is, because you know the hyphae, as a matter of fact, they grow whether inside the root or outside the root, but their hyphae, because it can extend, has a lot of surface area, and they're able to absorb a lot of nutrient, especially if the plant growing in an area with a low nutrient deficiency, especially phosphorus. So the presence of these are very important because they can able to absorb it and of course even send it to their host. Okay, and so these are extremely important because they are hyphae enter the living roots, okay, the living root of cells, okay, of perhaps, as I mentioned to you, okay, 90%, as I mentioned, of all higher plants. The introverted core of all higher plants. Of course, to mention one thing here, okay, and basically, these have basically form what is known as the mycorrhizae of higher plant, which form as mycorrhizae. We call it mycorrhizae, okay. Now, to continue of higher plant mycorrhizae, okay, and establish, yeah, and because I'm looking there for the time at the top of the board there, and establish, okay, With them means with the root, with them. Okay. Obligate. Obligate, mutualistic, which is basically obligate, mutualistic, symbiosis, which is we call it also another word, the mycorrhizae symbiosis. And this is basically called arbosicular mycorrhizae, known as arbosicular mycorrhizae. In other words, also it means endomycorrhizae. Endomycorrhizae. Okay, because here I have to stop my lecture. I just want to mention to you something here. Okay, because just a minute here. Okay, now usually in the mycorrhizae, just give me a second here. Okay, in the mycorrhizae, basically this is shows to you a cross section of a root that we have some fungi. They will grow outside of the root, like this one. This is known as ectomycorrhizae. This one here, they grow inside. And the reason why we call it arbosicular, because they go inside the cell and they form swelling like this. And that's we call them arbosicular mycorrhizae. Okay, by this one, I will stop my lecture here. We'll continue next lecture. And for those students who came late, because already we agreed to give the midterm test, is going to be on October the 30th, okay? And I'm going to send you an email by D2L to mention about that, okay? Well, have a good day because I think very shortly it's going to close. Okay? Did you for lecture okay?
Yes.
Thank you.
Yes, thank you.
Thank
Thank you,
you.
Professor.
Thank you. Have a nice day.
Well, for you. Bye-bye.